Okay, hi everybody, this is Lilith and I'm one of your IAs for BIBC 102. In this video, I'll do an overview of Lecture 10. So before we move on, I wanted to give you an overview of what is happening in the mitochondrion, in hopes that this will put the big picture together for you all and help you in your understanding of all these processes. So over here, we have the inner membrane of the mitochondrion. Not pictured in this picture, there is also an outer membrane. The space between that outer membrane and the inner membrane is called the intermembrane space. And the space inside of the inner membrane is called the matrix. For reference, glycolysis takes place in the cytosol, which is outside of that outer membrane, right? Cytosol would be out here. Oh no, malfunctions. So if this is the outer membrane, cytosol would be right here. Okay, then after glycolysis, the product of glycolysis, pyruvate, makes its way into the matrix of the mitochondrion, where it gets turned into acetyl-CoA that enters the Krebs cycle still in the matrix. Okay, during that time, the Krebs cycle produces various molecules of NADH, one FADH2, and a GTP that later turns into ATP. Okay, now you may ask yourself, well, what happens to those electron carriers? Well, what they do is they travel towards the inner membrane, right? And why do they do that? Because there are these complex proteins embedded in the membrane that work together and work sequentially at the end to produce water and ATP. Okay, so that's the big picture, right? We have the electron transport chain. What it does is it takes electrons off of NADH and FADH2. It transfers the electrons from one molecule or protein to another, and it does it enough where the last electron pair ends up on molecular oxygen that we breathe, turns that oxygen into water, which is abundant in every cell and every organism. And then at the end, somehow, right, you don't know how yet, but we'll talk about it, somehow an ADP gets phosphorylated into ATP. Okay, so that was the very simplified version of ETC and how it works. So let's dig a little deeper and actually talk about how each one of these functions and what happens. So to move on, I just mentioned that electrons are able to get transferred from one molecule to another, right? And for that to take place, these proteins, right, or these molecules must be able to carry these electrons, right? And by carry, I mean either by holding on by covalent bonding. For example, you know how in NAD, if we add protons and electrons to it, then it becomes NADH, and in the structure, those hydrogens are actually making a covalent bond, and that bond includes those electrons that the NAD is carrying. Okay, hopefully that makes sense. So let's talk about those electron carriers that exist within the ETC. And in class, we talked about 3 of them. So one of the key electron carriers in ETC is ubiquinone, also known as coenzyme Q. And for reference, this little circle over here is coenzyme Q. It's not actually a circle, it's just depicted as a circle. Or maybe its shape is globular, I'm not sure, but this is coenzyme Q. And how does coenzyme Q carry electrons? Well, let's take a look at its structure. So coenzyme Q has these ketones on both sides. And what happens is this form is fully oxidized, right? This molecule can't get any more oxidized. So let's see what happens when it is getting reduced. So first, we add one electron to it and a proton to go with it. And what happens is there's some electron pushing, right? And this electron with the proton ends up on one of the ketones, making it an alcohol and making this inner cyclic structure a benzene ring. Then what happens? A second electron comes in and attaches to this radical right here and makes another alcohol group. Okay, so now you can see how this molecule that was fully oxidized is now fully reduced because we've added 2 electrons to it. Okay, so that's coenzyme Q, and that's how an oxidized version of coenzyme Q gets fully reduced. The next molecule that acts as an electron carrier are a type of cytochromes, right? So cytochromes are special in a way where they have this iron group in the middle. And what iron can do is it can bind electrons. And what cool is about cytochromes is that they also can change their colors due to oxidized or reduced forms. This structure is present in cytochrome C, which is over here in the electron transport chain for reference. So we've so far talked about coenzyme Q and cytochrome C. And another way of electron carrying in the electron transport chain is having your proteins have sulfur and iron groups. And this is also another way of carrying electrons because these groups can carry electrons themselves. Okay? And so this complex is in proteins. So several of these proteins contain sulfur and iron complexes. Okay? Hopefully that makes sense in a way that each of these proteins and molecules has its own way of carrying electrons and passing it on via oxidation and reduction reactions. Okay? Now, the basic idea of electron transport chain is to transfer electrons from one carrier to another while releasing small amounts of energy at a time. The other purpose of having an electron transport chain is to make the electrochemical gradient across the inner membrane of the mitochondrion to later be able to drive ATP production. Now this green part you'll understand a little later, but I want to explain this a little further. So let's go back to this picture. So I'm trying to find a good way of explaining how oxidation and reduction takes place in the electron transport chain to be able to move the electrons this way, because we ultimately want the electrons that are here to go this way and come down on the oxygen so we can make that water. So think about it this way. NADH comes in and it has 2 electrons carrying, right? These electrons get put on complex 1. Then complex 1 takes those electrons and gives them to coenzyme Q. Now coenzyme Q has those electrons, and coenzyme Q gives them to complex 3. Now complex 3 has these electrons, and it gives them to cytochrome C. Cytochrome C has those electrons, gives them to complex 4. And finally, complex 4 takes those electrons and gives them to oxygen. Now the word gives is very colloquial, right? And hopefully that gives you an idea of how exactly the electrons are transferred. But what does gives mean? It means that when NAD is in NADH form, it is reduced, right? Because it has those electrons. Now, complex 1 initially is oxidized, meaning it does not have any electrons. So when NADH comes in with those electrons and reduces complex 1 by giving complex 1 those electrons, complex 1 now becomes reduced with these electrons. Okay? Now, the next step is coenzyme Q. Coenzyme Q is oxidized because it doesn't have any electrons. But guess what? Complex 1 does, because it is reduced. So what complex 1 does is it reduces coenzyme Q with its electrons. In turn, complex 1 becomes oxidized because it just gave up its electrons. And coenzyme Q now becomes reduced because it just gained 2 electrons. Then the same thing happens. Complex 3 is oxidized because it doesn't have any electrons. Where can it get those electrons? Well, it can get reduced by coenzyme Q, who has those electrons. So coenzyme Q reduces complex 3. Coenzyme Q now becomes oxidized once again. And now complex 3 is reduced because it just gained those 2 electrons. Now cytochrome C is oxidized. Hopefully by now you understand why is it oxidized? Because it doesn't have any electrons. Now complex 3, because it is reduced itself with those electrons, it reduces cytochrome C. In turn, complex 3 becomes oxidized because it just gave up its electrons. And now cytochrome C becomes reduced because it just gained those electrons. Now, complex 4 is oxidized because it doesn't have electrons. So cytochrome C, being reduced, gives its electrons to complex 4. Cytochrome C itself becomes oxidized again because it just gave up its electrons. And now complex 4 becomes reduced with 2 electrons. And lastly, cytochrome 4 reduces oxygen, right? And oxygen is initially oxidized. So when it gets those 2 electrons, it becomes reduced into water. Okay, so hopefully that oxidation reduction play made sense. So let's move on. Now, all of that I said, that we already know, right? And why do we know that? We know that because many other scientists and biochemists at the time did a bunch of experiments and procedures to find out how exactly the electron transport chain works. So we talked about 3 experiments or concepts that previous researchers used to understand how exactly ETC works. So the first experiment or concept that we talked about in class is the order of ETC being explained by half-cell values, right? E naught. If you remember, the more positive the E naught is, the more prone it is to getting reduced. And the more negative the E naught is, the more prone it is to getting oxidized. Remember that reduction is getting electrons added to you. So species or molecules that are prone to getting reduced, they want electrons, okay? But molecules that are prone to being oxidized, they don't want electrons. And even if they have electrons, they want to give them up, okay? So with this in mind, researchers looked at the E values of the molecules and proteins in the ETC. And they figured out that there is some kind of order and some kind of sequential way that they can order these complexes. And it turns out that if you put them in increasing values, so becoming more positive, then you get this exact order that ETC is in. And what does this tell us? This tells us that the most positive E naught value is the last reaction. This means that oxygen really wants to get reduced, right? It is prone to getting reduced because it has this positive E naught. The reaction before it is not as positive, right? This is 0.8, this is 0.3, so it's not as positive. That means this is not as prone to getting reduced. It is actually more prone to getting oxidized. Then going back one more time, this value is even smaller. This one is even smaller, smaller. So you see how from the top to the bottom, numbers grow, meaning as you go from this complex to this, and then from this to this, and then from this to this, and from that to that, the reactions become more and more prone to getting reduced, meaning they become more and more prone to wanting to get those electrons. So by understanding this order, researchers understood why exactly every electron transport chain embedded in the inner membrane has the exact same order. It's always complex 1, coenzyme Q, complex 3, and 4, and then oxygen turning into water. One reaction that I missed is complex 2. And complex 2 does what basically complex 1 does. Complex 1 takes electrons off of NADH and gives them to coenzyme Q. And complex 2 takes electrons off of FADH2 and gives them to coenzyme Q. Where does this FADH2 come from? It comes from succinate getting oxidized to fumarate. In the process, you make FADH2. That later is used by complex 2 to reoxidize into FAD and take its electrons. Okay? Now, going back, This was the first experiment, or one of the first experiments, that identified and explained the order of these complexes in the electron transport chain. The next experiment we talked about is understanding the chain of ETC by using inhibitors. Now, we've talked about inhibitors before, right? If we take an inhibitor and we inhibit an enzyme, what happens? The substrates of that enzyme accumulate. They increase in number, right? Because they're not being processed. And the products decrease. So by knowing that these molecules get reduced as you go to the right, then what researchers did is they took inhibitor molecules that inhibit specific enzymes in this chain. So when you inhibit this, you can tell that whatever's before it is already reduced, but whatever comes after it is still oxidized. If this was not inhibited, right, if this was left alone or just activated, then this would be reduced, this would be reduced, right? Everything would get reduced all the way to oxygen becoming water. However, if you inhibit these enzymes, you can tell how everything before it is already reduced, but not the ones after it. Okay, and the last way of figuring out how these enzymes and complexes work is to simply do a lot of biochemistry, right? And I've drew celebratory lines around biochemistry because this is a biochemistry class, but also I'm biased because I love biochemistry. But basically what scientists did is they took the mitochondria, right? They lysed it open. So lysis is to break the organelle, right? They broke the cell membrane. They harvested the cell membrane or inner membrane patches that contain these separate complexes, and then they separated out each of those into different tubes, and they studied them separately. So they gave them a substrate to see what happens, right? They looked at color changes, and they performed other biochemical techniques and procedures to examine all of these proteins individually. Okay. Now that we know all of that, let's take a look at the full picture once again, and let's talk about this whole electrochemical business and what this is. Okay, so over here we have complex 1, complex 2, complex 3, and complex 4. In the middle over here we have coenzyme Q and cytochrome C. So one more piece of information that I have yet to tell you is that complex 1, 3, and 4 are actually proton pumps. This means that they can take a proton from the matrix and pump it out into the intermembrane space. As you can see, I've added to this picture, I've put a lot more hydrogens outside over here and very few in the matrix. Now, these proton pumping functions are against the hydrogen gradient, right? If there's a lot of protons over here already, no more protons want to actually go up here. But because we have these pumps, right, these pumps forcefully take out the protons in the matrix out into the intermembrane space. And that is important. That needs to happen to give us this gradient that is going to be very, very useful at the end. Okay? By making this forced pumping of protons, we make what's called an electrochemical gradient. And I've highlighted electro and chemical separately because they mean different things. Okay, there is an electrical gradient along this inner membrane. And why electrical? Because we have a bunch of positive charges in the intermembrane space and not that many positive charges in the matrix. So relatively, the intermembrane space is way more positive than the inside. That's why we sometimes call the intermembrane space the P side for positive, and the matrix side the N, which is for more negative. Okay, so that's an electrical gradient, more positives here than less positives here, so more negatives in the matrix. The chemical gradient is actually having a bigger concentration of protons in the IMS than in the matrix. And this is important as well, because if we have a lot of these protons out here, a lot of these acidic hydrogens, then inside here, then you can see how that is a gradient also, right? For people who haven't heard the word gradient before, I'll go over it very quickly. I just realized that not everybody might know, so I'll just zoom in over here and explain. So think about a room, okay? And the room is full of lots of people, right? So let's say we take 20 people and we put them in this room, right? It's a small room, 20 people, everybody's just closely packed in that room, right? Not safe for COVID reasons, but in a hypothetical situation. Let's take 20 people and put them in this very tight room. They are not going to be happy, right? They're wanting to go out, but we've closed all the doors, so they're just going to stay there. But as soon as we open one door, right, everybody will come out. Everybody will rush out, right? Because they don't want to be near each other anymore. It's too tight. These other people are driving each other crazy, right? They will run out. This will be a gradient of people, okay? I'm not sure if this analogy is working. But basically a gradient is when you have a lot of something in one area, okay, there is a difference in the amount of that thing in that area. There's a difference between that and somewhere else. Okay, so there's a lot of people in location X and not a lot of people in location Y. And that's a gradient. So when there is some kind of opening, then the gradient will equalize because the lots of people in here will rush out into location Y. And eventually there will be an equal amount of people in here and in here. Okay, the gradient will equalize. Okay, you also might have heard of the word osmosis. And osmosis is the movement of water from one place to another. So let's say we put a cell in a very watery solution. So there's a lot of water molecules in this solution, and there's only very few water molecules in the cell. What will happen is these water molecules will rush into the cell, and the cell will swell up. Okay, so that will be osmosis, the movement of water down its concentration gradient. Okay, so hopefully that whole gradient talk made sense. I will go ahead and erase this. Sometimes my analogies don't make sense, so hopefully this one did. But basically, in the inner membrane space, when we have this storage and packing of protons, there's not a lot here. So if there is some kind of opening somewhere, you know that all of these protons are going to want to rush inside the matrix. Okay? And that's exactly what the ETC wants to do. It wants to make a situation where the protons will want to come in. Okay? And why do we want to do that? Well, because that's the main way of making ATP. What you'll notice is that right next to the ETC, there is this huge multi-subunit protein called ATP synthase. And what ATP synthase does is it has a portal inside of it that is an opening back to the matrix. So if there's a lot of protons in the intermembrane space, they're going to be super happy because they're going to say, look at that, there's an opening. And they will rush back down into the matrix because there is this electrochemical gradient across this membrane. Once these protons rush in, they will produce enough energy to take ADP and phosphorylate it into ATP. Okay, so that's an easy way of understanding how ATP synthase works. Okay, now that was an overview. I know in lecture, Professor Hampton hasn't gone over exactly how ATP synthase works, so we'll look at the details this week. But it's good for you to start thinking about how that electrochemical gradient across this inner membrane eventually leads to ATP synthase, making ATP. Some key words that you want to think about that you will learn this week is chemi-osmotic hypothesis. There's a few familiar words in here. Chemi for chemical, osmotic for osmosis, right? Moving chemicals down their concentration gradient. Proton motive force, right? The motivation that protons have to move from up here to here, down their concentration gradient. And then oxidative phosphorylation is how ADP gets phosphorylated into ATP. Okay, we will learn more about this week, and we will go over the details much better in the next videos. So hopefully this gave you a quick overview of what Lecture 10 was about, and if you have questions, please do email me or come to my office hours. My name is Lilith, and this will be it for this video. Thank you.